Okay, now we're going to talk about the actual structure of DNA, and some of this might be a little review from some stuff we talked about earlier in the semester. So DNA is going to be a polymer. So if you remember, a polymer is going to be made up of repeating subunits called monomers, and the monomers are going to be nucleotides. So if we go to the PowerPoint, you can see right here that this is going to be a nucleotide. So a nucleotide is going to be composed of a phosphate group, a 5-carbon sugar that we're going to get a little bit more into in a second, and then the nitrogenous base. So those are going to be the 3 parts of a nucleotide, and a nucleotide is the building block of DNA. All right, back to the notes. The nitrogenous base that was over to the right in that picture can either be what's called a purine or a pyrimidine. Purines can either be adenine or guanine, and we're just going to call them A and G. And pyrimidines can either be thymine, cytosine, or uracil. Uracil is going to be something that we only find in RNA, and thymine is something we only find in DNA. So those will come up again a little bit later. Okay, so when we look at that 5-carbon sugar again, let me pull this up. We're talking about this right here. And so remember how I told you a while ago that every point in a drawing like this is going to be a carbon. So if we are talking about the different carbons in the sugar, we're going to want to label them so we can kind of just know right off the top of their heads which carbon we're talking about. So if you look here, we have carbon number 1 right here, carbon number 2, 3, 4, and 5 is up here. Now, if you notice, they have a little apostrophe next to them, and that is standing for prime. So we say 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And all that is letting you know that we're talking about the carbons in the sugar and not in the nitrogenous base. Because if you look here, those could all be carbons too. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. All right, now if we look at this part of the picture over here, you can see that off of the 3' carbon, there's going to be an OH. Off of the 5' carbon is where the phosphate group is, so you can see here. So what's going to happen is, remember we talked about how monomers are going to be joined through the process of a dehydration synthesis. So what's going to happen is we can keep having a dehydration synthesis and adding one more and one more and one more, and we can just keep adding and adding and adding. However, what I want you to be aware of is that when we make this chain, there's going to be an exposed 5' end where the phosphate is up here. And down here at the bottom, the end that's exposed is the 3' carbon with the OH coming off of it. Okay, so even as the chain gets longer and longer and longer, it's always going to have an exposed 5' end and an exposed 3' end. So that's really important to remember, because when we start to talk about how DNA replicates itself, that's going to come into play and cause all sorts of problems, because why would it be so easy, right? All right. So this is all talking here about the 1', 5', and how we have all of the different things coming off of each end. Now the type of bond between these guys is going to be a phosphodiester bond. Now what I mean by that is, if we go to this, you've seen DNA, and DNA can be drawn like this, right? And then it looks like a twisted ladder. All right. I'll switch colors here. If we are talking about the bond from here to here, that's a phosphodiester. That is the longest word ever created, bond. So this bond from one nucleotide to the next is a phosphodiester bond. That's going to be different from the bonds here to here. Those are going to be hydrogen bonds. Okay? Wow, I can't spell well today. All right. So all of the rungs of the ladder are hydrogen bonds. All of the sides of the ladder are phosphodiester bonds. So that's something you want to keep in mind, because people tend to get confused with that. Okay, back to this. What's going to happen is we're going to refer to nucleotides always in the 5' to 3' direction. Now, what do I mean by that? Let me clear this off and I can show you. All right, if we were going to draw our DNA like this again, ooh, that was really bad. You get the idea. Okay, if we were going to look at a close-up of one of these strands, let's say that we wanted to look at a close-up of this strand here, it would look like this. It would be like A, C, G, C, T, 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 A, C, whatever, right? So that's talking about the actual nucleotides that are making up one side of this ladder. And everywhere you see a line, that would be where you'd have another letter, okay? So let's say this end is the 5' end and this end is the 3' end. We're always going to talk about it in the 5' to 3' direction. And the reason for that is because that's the way that most of the enzymes that we're going to talk about are going to read DNA. So they like 5' to 3', so that's just kind of like a rule that we're going to have, okay? Now, if we go over here, we need to talk about Chargaff's rules. Now, Chargaff's rules are talking about the A's, T's, G's, and C's. Now, I should mention, why aren't we talking about the letter U? Remember, the letter U is only going to be in RNA. So right now we're talking about DNA. So he was the one who figured out the amount of A always equals the amount of T, and the amount of G is always equal to the amount of C. So that started some experiments to say, is there a relationship between A and T, and is there a relationship between G and C? Because it's kind of crazy that they're always equal to one another. So there's always an equal proportion of purines and pyrimidines. Now, another scientist, oh, poor Rosalind Franklin, she came along and she was actually using X-ray diffraction. And the reason I say poor Rosalind Franklin was because some crazy things did happen to her. Let's see. So this is an actual picture of the X-ray diffraction that she used, and she actually was not as famous as she should have been, but she was the one who came up with that helix shape, right? So it's like the twisted ladder shape. And she could actually figure out that it was 2 nanometers wide, and then it made a turn every 3.4 nanometers. That's in 53. I think that is super cool. Now, the reason I say, you know, poor thing is because there are some rumors that Watson and Crick stole some of her ideas and presented them as their own. The other sad thing is she did win the Nobel Prize, but she won it, I believe, you know, after she was dead, and she died due to all of the radiation exposure she got from the X-ray diffraction. But anyway, we can look that up a little bit later. All right, let's talk about Watson and Crick. They were the ones, so basically if you want to think about what Rosalind Franklin came up with, she came up with the shape of it. So like as if you took your shoelace and you twisted it, she could figure out that much. These guys actually came up with the fact that there are actually 2 chains of DNA that are intertwined. So a little bit more detail into what she came up with. Now, complementarity is another thing they came up with. And what I mean by that is they figured out that A always goes with T and G always goes with C. So if we're looking at this strand that I drew here, everywhere there's an A, there's going to be a T across from it, T. And everywhere there's a C, there's going to be a G. Everywhere there's a G, there's going to be a C, G, A, T, right? So this is our strand, a double strand of DNA. So you can see how those go together. That's complementarity. So A is always going to go with T, G is always going to go with C. The other thing they came up with is something called anti-parallel configuration. And what that means is if one strand is in the 5' to 3' direction, the other one is going to be 3', 5'. I'll show you again on my little thing here. Let me just clear this. So what I mean by that is if we have, we'll go sideways for this, a strand of DNA, and I've just straightened it, right? So it's just not looking like a twisted ladder. If this strand up here is 5', 3', then this one will be 3', 5'. Now, why am I mentioning this? Because this is going to cause huge problems when we start to talk about DNA replicating itself. So that's called anti-parallel configuration. Now, in the next video, we'll get into how DNA actually replicates.